This time we're going to stay in the same galaxy of cancer systems, but a totally different solar system, as it were. We'll also touch on machine learning, statistics, and large-scale data. Our passionate speaker is Dr. Kasturi Kannan, an associate professor and director of Computational Pathology program at the University of Texas MD Anderson Cancer Center in Houston, Texas. He's a data scientist with an expertise in cancer genomics, epigenomics, biomedical informatics, and image processing. Kasturi is also working currently on systems biology and spatial stochastic processes with its application to multiplexed immunofluorescent images. I'm not even sure what I just said, but I'm sure it'll all be clear in the next 30 minutes or so. Kasturi, the stage is yours. Thank you, sir, for being here.
Oh, thank you, Holmes. This has been a wonderful gathering, and it's my pleasure. I have known Ilya for quite some time, and I'm nice to meet him and everybody here. So let me start sharing my screen. Are you able to see my screen?
We can see your screen, but you might want to go to, there you go. Absolutely great.
All right. Okay, so let's get started without any further ado. Today, I'm going to talk about bioinformatics and cancer systems biology. In particular, I'm going to touch upon why bioinformatics has been critical for cancer systems biology. I have nothing to disclose or no conflict of interest. Before going into the topic, first of all, I would like to establish my credentials on this area in general about what led me so far to the current position here. Basically, I'm a mathematician. I got my master's in mathematics from India, and then I moved on here to study mathematics. In particular, I was more interested in topology, approximation theory, etc., etc. But my interest shifted towards computer science, and I graduated with a PhD in computer science, especially in computational physics. Until about 2008, I have no idea of what bioinformatics is or biology or bioinformatics. I should share an anecdote that, in fact, my mentor, she wanted me to pursue bioinformatics, and she was interested in protein folding. And at that point, I said, no, I'm not at all interested in bioinformatics. But it later turned out that I went to a medical research institute where I got some basics of biology, and that led me naturally to bioinformatics, working with the data sets, biological data sets. And I was doing genomics, large-scale systems, studying brain tumors at Memorial Sloan Kettering Cancer Center. And then that led me to the position at New York University, where I was an assistant professor. And I came to know about Ilya, and we had a good conversation, and I got introduced to him at that point. And slowly that led me to a current position at MD Anderson Cancer Center. I am an associate professor and director driving majorly what is known as computational pathology. But that's not going to be the topic of today. I'm going to more talk about another exciting area that I've been working for about maybe 2 years now. In general, my expertise it's varied. It's like a Swiss Army knife. In general, I would like to call myself as a data scientist. About 15 years, specifically focusing on cancer research. All right, so let's go to the topic. Let's go to the state of modern biomedicine. Right? It's like a big elephant, and we all know the story. That elephant and blind men, right? Some people touching their tails and thinking that's a snake, and some of them touch the skin and saying it's a wall, etc., etc. But nobody gets a big picture, right? It's so fragmented, and this fragmentation actually appears, especially when it comes to drug discovery. We have water everywhere, not a drop to drink, and data everywhere, not a drug. I think that's one of the famous quote that's been widely used now. But the key thing is that we all know that the medicine is very, very fragmented and bifurcated. And so we are having a hard time in reconciling, what directions to pursue, what are the good targets. People go by single cell RNA-seq, or all the things have now come to the fore. A good thing is that we have discovering so much, the data is everywhere. But unfortunately, it's all difficult to put everything together to find a bigger picture of what's happening in modern biomedicine. So the key question as we dive into the topic is about nature versus nurture, right? At the end of the day, we can think of genetics as something hard-coded in our DNA, right? Mutations, deletions, etc., etc. And then there is a whole phenomenon of epigenetics where the methylation and other things can contribute to gene expression silencing, and we know that this is another major mainstream mechanism. So people are like, some people subscribe to genetics and some people subscribe to epigenetics, like I was telling about the elephant and blind man, right? I mean, I have seen people saying that, okay, I am a geneticist and I believe in epigenetics. So the key questions is how they interplay, right? One of you, I mean, whoever have a strong base in studying the DNA might subscribe to the DNA model, the basic central dogma, and say that genetics is the key thing that's driving, but we really don't know the answer. That's the truth. So in the absence of consensus, right, in God we trust, all others must bring data. So what does that data tell us, right? Whether it subscribes to genetics or whether it says that it's genetics and epigenetics, we know that it's both, but how much of it, right? I mean, the problem is that the data, now we have a deluge in biology, right? Explosion of data sets. Now we have the NGS, you're all bioinformaticians, you know about all these things. I don't have to tell. But then the primary aspects of the data are mutations, copy numbers when it comes to DNA. Of course, gene expression could be controlled both by the DNA as well as the RNA. And there are so many other things like structural variants and so many other things that we could study that we are unable to get a consensus of what actually is driving, right, the tumors. So yeah, I mean, it's like studying the wings, right? And saying that, oh, the aeroplane just runs because of wings. And some people study the engine saying that, oh, it runs because of engine, right? Of course, we know that it's both, but how do we study both, right, without ripping the plane apart, right? How do we really study the entire structure, right? The aerodynamics is not just an outcome of just the wings or an engine alone, right? The aerodynamics is determined by the flow of the air, and it's a process, right? The dynamical process. It's a spatio-temporal process, and not a static thing that we could study by ripping things apart, right? So these are some of the things that I have been thinking for a long time, and that's when I hit upon the systems biology. And I've been thinking about systems biology. I've tried to watch some videos, and everywhere. When people talk about systems biology, there is not a definition of it, right? People are saying, okay, yeah, I mean, systems biology, it's tough to define, right? So the problem is because, let's forget the systems biology for a moment, but the whole point of just studying these mechanisms, the biological mechanisms, is that the modern biology is more or less reductionistic, right? We break it apart, we study, hoping that when we study these parts and try to put together, we will get a understanding of the entire mechanism, right? I mean, it is like saying that you have, let's say that we have a bunch of best cars in the world, right? I mean, one is good for, let's say, a BMW as a best engine, and a Volvo probably has the best transmission, and say some other car has, a best best battery or something like that. So when we are trying to rip everything separately and trying to organize something that we can put together, we will not even get an automobile. That's the unfortunate thing. We will be able to rip it apart, we will be able to study, but putting it together is not, it's possibly impossible. So the divide and conquer approach will not work when studying complex systems, especially biological systems. Especially cancer, right? Cancer is a very complex and adaptive. It's an evolutionary mechanism. The dividing cells constantly adapt to their environment, and so it becomes it's an adaptive system. And so we will not be able to put together and study how the mechanism works in real time, right? So when we think about how reductionism works, it is like a relational database, right? I mean, we break things into tables, and we study each tables, right? And then we hope to do some joins and understanding, okay, so I'm going to join this table X with table Y using some primary key or a foreign key, and hoping to understand table X and table Y together, right? I mean, these joins are made out of inference. It is like a reductionism. So you can actually think about reductionism as a relational database, but this is not going to work. because putting it together, it's just an interpretation at some point, right? It's not going to reveal an answer by itself. So we need an integrative paradigm, which is known as a systems approach. A systems approach is putting things together and studying it in a very comprehensive fashion. Whether are we ready there yet, whether are we, do we know how the mechanism works by integration? That's something that's the biology of the future. But the right approach would be to actually put things together to see that what makes sense and what doesn't make sense. The systems approach is characterized by a simple thing: the whole is not the sum of its parts. So that's the key sentence that defines systems biology. So the whole, the entire picture that you see, is not going to be the sum of the parts, because the interaction between the parts matters as much as the parts themselves. Without the interaction between the parts, the part doesn't make any sense whatsoever. So that is the entire idea behind systems biology. So how do we then define it, right? So I was looking into the definitions of systems biology, and I came into this Institute for Systems Biology, and they have this model where they say that, okay, you have the technology, and then new technologies give rise to new data, and then new data give rise to new software, and then out of that, doing some bioinformatics, we form new hypotheses, and then the hypothesis will gain rise to new insights, and then new biological questions. This is a life cycle of technology, computation, and biology that defines systems biology. But this is not true, because it's an integrative technology that defines systems biology. The integration, like I said, the whole is not the sum of the parts. Systems biology and complex adaptive systems are defined by the integration. And when you look at it, it's just the technology, just doing some next generation sequencing or any of these is not going to be, or imaging is not going to define systems biology. All we will get is a bunch of data. And of course, we can do bioinformatics with a bunch of data and then try to answer some questions, but that is not what systems biology is. So with this, we started to trying to think about the goal of the project. So I started thinking about, okay, what it makes sense, but I'm going to, before I go into the details of how we can really identify systems biology and what it has to do with bioinformatics, right? I was thinking of doing a project actually to define, to really showcase what it is, right? So the goal of the project is creating a unifying computational framework. So it's a computational framework, and we want to integrate all available patient data. Okay, so we have the tumor recurrence data, the primary data, and the recurrence data, and so we want to integrate all of them, and then in an unbiased manner for actionable target validation and predicting disease outcomes. So that's the idea behind the project that we initiated at MD Anderson Cancer Center. When I joined, I wanted to start the systems biology initiative for brain tumors. And the key thing is about integration, right? It's an integrative technology that we have to use here. So why brain tumors? Of course, it has been my expertise for a long time. For about 10 years, I've been involved in brain tumor research, both in neurosurgery and neuro-oncology. In neurosurgery, I hold a joint appointment there. So in general, I have been studying brain tumors a lot. And so that was natural for me to do this cancer systems biology in glioma. So gliomas are in general the nervous system neoplasms that is derived from epithelial cells. In general, the differentiation or classification is by glial cell types, right? I mean, usually the brain cells, if you take, there are neurons, there are astrocytes, oligodendrocytes, and epidermocytes. So in general, the gliomas arise from glial cells, and it constitutes about 30% of all central nervous system tumors. And in fact, in particular, if you look at the brain tumors, we have about 70% of them are brain tumors or gliomas. So they are divided into two major categories: the low-grade glioma and the high-grade glioma. Usually, the low-grade gliomas are grade 2 and 3, which largely consists of astrocytomas, oligodendroglioma. And then grade 4, which is a high-grade tumor, it's usually known as a glioblastoma. It is one of the most lethal cancers. People die in about 14. Invariably, they die in 14 months. It's a very aggressive tumors, and no cure has been, or no reasonable progress has been made on gliomas, in glioblastoma for the last 40, 50 years, despite there is little progress in terms of chemotherapy and stuff like that. There's not been much progress on that, right? So we not, and then the TCGA, the Cancer Genome Atlas, started with understanding of glioblastoma as one of the key projects in there. And so we know quite a lot about molecular biology of gliomas from a DNA, RNA, and protein and epigenetic levels, right? We know a lot through the sequencing, doing bioinformatics. We have tons and tons of data now in TCGA on glioblastoma, right? But then the problem is that people invariably recur, right? Even though they are given chemotherapy, at some point they get the tumors again, and so we really don't know about what happens, how the relapse happens, and so we thought there should little is known about tumor, primary tumor and recurrence, about progression, resistance, metastasis, and so many other things. So that's why there is something called glioma analysis consortium, called the GLASS consortium, was formed to understand glioma from a recurrence and a progression perspective. And so the GLASS aims to perform a comprehensive molecular profiling of matched primary and recurrent tumors from 1,500 patients. And so this consortium includes investigators from 14 academic hospitals and universities. MD Anderson is one of the core sites for this GLASS consortium. And this GLASS consortium, when I joined, I'm a part of the GLASS consortium. It's led by investigators in here. So I thought the best case for defining a systems framework should be to do a GLASS, use the GLASS data that has already profiled tons of tumors, both primary and recurrent tumors, and study how the tumor progression is happening in terms of both bioinformatics and systems biology perspective. So what we know about recurrent gliomas is that the first published study, they analyzed about 222 patients with glioma. So they analyzed mutations, copy numbers across a variety of molecular subtypes of glioma. And there are three major molecular subtypes of glioma. One is IDH caudal, IDH non-caudal, and IDH wild type. So what we know about is that the study also said that, okay, driver genes are initially selected and then they were retained at recurrence, but there is little evidence of recurrence-specific alteration. So most of them happen at the primary stages. And these are some of the inference that they have derived. The alkylating agents introduce what is known as a hypermutator phenotype. And then aneuploidy, abnormal number of chromosomes, is frequently detected in recurrent gliomas. And then the clonal architecture of each tumor remains similar over time. And no difference in the level of, from the immune standpoint between initial and recurrent tumors, there's no difference. And these are the inferences that was derived. But the thing is that we have to understand these are done just using the basic bioinformatics. They have not done any data integration across several types of data to understand glioma recurrence in a very integrated fashion, right? So that's the key thing that we wanted to study again. What is missing is that the key thing is that what they have understood from doing some rudimentary bioinformatics is that it's largely stochastic, right? But then we have to integrate gene expression data, we have to integrate methylation data, clinical data. Think about the initial, the elephant and the blind man slide, right? I mean, we did some things separately. We did gene expression analysis separately. We did DNA mutation separately, methylation separately. But then we have to put it together to understand it. And how do we do that, right? That's the goal of the project. We wanted to create a unifying, again, to reiterate what we wanted to accomplish is that creating a unifying computational framework to integrate all available patient-centric tumor recurrence data to generate hypotheses in an unbiased manner for target validation and predicting disease outcomes. So this is the systems approach, not the regular traditional individual bioinformatics approach, right? So how do we do such an integration? So here comes the meat of the idea of how are we going to integrate, say, very disparate data, let's say the gene expression data with mutation data or methylation data with copy number data. How are we going to even integrate all of these things, right? So that's where what is known as graph databases come in, okay? So you might all have a known graph. By graph, you might have meant a pie chart or some kind of a pictorial representation, but that is not what a graph is. From a mathematical standpoint, it's a name for a network, right? It consists of nodes and edges, right? So in traditional databases, you have these separate tables, and we are trying to infer things together, right? So you have mutations. Let's say you have a copy number table and a methylation table, and you join them, try to do some understanding based on the mutations, copy number, and methylations would be done based on some bioinformatics, right? I mean, there are mutation callers, some pipeline, GATK pipeline, and so many other things, right? But then there is there is no explicit relationships that are embedded into these tables, and so you have to infer these things separately, right? But graph databases, the relationships are defined, right? Think about Facebook, think about Amazon, think about all these things, right? I mean, how do they even work? For instance, Amazon, you buy a product and immediately says that, okay, this product what do you mean? When you buy this product, people who bought this product also had brought another product. So how do they infer that in a real time, right? There is no person sitting there and trying to analyze your product and then try to match up with other people's product who also purchased the product, right? I mean, these relationships are already embedded in here. And so it's just a matter of query, right? There is a system already in place that has these relationships defined. And so it immediately points out to the product people who bought this also bought this. It's an association, right? Those associations are already mapped. Same with things happens with Facebook, right? A friend of a friend. Friend is a relationship between two people, right? So when you have is a friend of can be embedded so that it can be naturally rendered in real time. And similarly happens with Twitter. And also many new technologies are just based on these kinds of graph databases. So the graph architecture, the integration is natural, and it's an unbiased analysis becomes very, very easy, right? So all you need to do is define the relationships, and so we can write appropriate queries to answer that, right? So is this idea new? Not really. If you think about this idea, it's already been one of the papers that was actually a very good paper. In fact, that was key for me to develop this. Is that they have already done this in what is known as a colorectal cancer. They had these mutations and that they have these relationships defined. And then they have tried to integrate all these together using the graph databases, right? And so the problem with their model is that they had to create additional relationship for new data. Let's say that you're introducing image data. So they have to introduce new relationship type. And also, it results in restricted survival analysis. We will go over that a little bit later. So then there are problems with the existing approach. The key attributes of the system that it should be integrative. You have to put in all data that you can find, and it has to be unbiased, as well as it has to be absolute in time. So these are the three characteristics of the system that we have to develop. And so, I was thinking about it, and the idea is very simple. So you have two molecular events, and you connect them, saying that if there are patients who undergo those events, then that forms a relationship. So the patients are the relationships in our model, right? So in our database, there is this basic representation. There are these molecular events, and you connect them if there are more than one patient who undergoes those two molecular events, right? So with that, we built this graph database where we were able to integrate copy numbers, where we were able to integrate methylation, mutation, expression. And if you are having imaging, we can integrate whatever data that you can get, we can actually put them in this database. So this is in this database, we call it as Graph Engine for Systems Medicine, or otherwise Genome. In this database, they have in this database, it's an integrative database. And so we would be able to query anything that we want, right? So there are 370 patients in this database so far, about 11K nodes and 16 million relationships. And this is the Venn diagram of how the distribution of the data looks like. So why bioinformatics critical? Now that comes to the critical question for this, right? So like I said, garbage in, garbage out. Right? I mean, it depends on how you define the system. And it's very important when you are constructing such a systems biology approach, it makes sense that we have to make sure that we really put in all the things that are important, right? So we make sure there is a good bioinformatics that is built in developing this system. So for instance, identifying mutations, right? We identify key driver mutations. So there is a pipeline already. we have alignment and GATK variant calling, and then we do the driver passenger analysis and do the mutation, driver passenger mutations, and then we upload it into the database. So it's important. Otherwise, it's all garbage. There's going to be millions and millions of data points. So this is where the bioinformatics is critical. Before it's the next, the whole systems approach is the downstream of bioinformatics. That's critical to understand. And there is, secondly, there is this patients. There is another filter in there. It's determined by the number of patients who undergo the molecular events. And also we have a robust statistics and connected events as another filter. And then we do a random walk and survival correlation. So these graph databases have a survival correlation. So like I said, right, two events are connected by patients. So a patient trajectory A could be different from patient trajectory B, could be different from patient trajectory C. So they will have different types of survival. So in general, we could enumerate all type of paths that's going to give different survival, but that's going to be an extremely hard problem. It's an NP-complete problem in computer science. So we will not be able to do that. So what we do is that we simulate a random walk. Random walk is like how many, it's just a tossing the coin. So if it's like, say there are 6 paths in here, if you throw a die and if it says 5, you take path number 5, etc., etc. So in general, we would be able to simulate a random walk and then do the survival correlation. So for instance, in this, one of the walks in here, we have the path number 51 and path number 54, right? They show the survival difference between the patients. And what you see is a bunch of different biomarkers or genes. That's a combination, right? It's not just a combination of it's just mutations or copy numbers. It's a combination of these that gives the difference in survival. So in this way, we would be able to determine the survival of patients. And this is another random walk simulation too. So we have these 2 paths which show completely different survival times. This is random walk 3 for another specific type of glioma, where we see the survival difference between the patients for path number 16, patients who take that path and patients who don't take that path. So the power of this approach, we have identified several functionally important genes for conducting biological experiments. So for instance, there is a recent study that says that glutamatergic synaptic signaling, it's important, reelin signaling. So we have identified all of these things. In general, right, this provides a strong framework for systems biology paradigm in medicine. It's patient-driven than patient properties-driven. It's truly unbiased and integrative. It's not subject to time. As long as there are patients, we are going to have this framework, and it's a correlating with survival, combining multiple markers has not been attempted. So with that, I would like to acknowledge the support I have got, the patients and their families. My student, PhD students, he has been actively developing the database. Dr. Hughes, Puduvalli, and then Dr. Lang, neurosurgery chair and neuro-oncology chair, they provided an unwavering support. Of course, my senior vice president and CTO of MD Anderson, he was like totally in sync with these ideas and helped us in providing all the support in here. Moonshot gave us the funding. Brain Tumor Center gave the resources. Of course, the database support at MD Anderson and the IT staff, we were available anytime that we wanted to go and had some problems. And of course, this was done on Neo4j and Neo4j team. Rob Martin, Fanny, and Greg Shirley, they were critical in providing the Neo4j for us in developing this system. So with that, I would like to end my talk and open the floor for questions.
Very good. Thank you. Thank you, Doctor. We do have several questions. If you want to go ahead and pull your screen off, we can just see you instead.
Oh, sure.
So one of the questions is, we'll even make you get me off the bottom here. There we go. So is the database you're describing being used by clinicians right now?
Yes, they have been utilized by clinicians. We are testing out certain biomarkers that were derived out of this database. Yes.
Okay, fascinating. So how important is it for a clinician to be aware of the data analysis methods to use it properly?
We wouldn't expect too much insight. I mean, for clinicians to understand the database and the queries, which we will handle. I think now what we would expect for the clinicians to know is that we can provide some actionable targets, and for them, if they understand a little bit of biology, to see that what makes sense and what doesn't make sense for them to conduct some biological experiments. I think that would be fantastic.
Okay, 'cause it's probably really important to know where and how the data was accumulated so they can use it properly. Is that right?
Yes, of course. Yeah. Yeah. I mean, yeah. So we have to develop systems like this for each and disease types. Yeah.
Thank you. Do you see any challenges that need to be addressed to integrate data when it does not come from the exact same patient, but rather different individuals?
Oh, yeah. I mean, this database is about different individuals, right? It's about population. So it's not just one patient. We have about 370 patients in our database, data that we are integrating. So yeah, any type of data, any type of patients, we should be able to integrate this database using this approach.
Well, thank you. Thank you for your remarkable work that you're doing with cancer. I loved all the metaphors throughout the elephant and all that. That's pretty good stuff. We're going to